Hi everyone. Tonight's video is on exceptions to Mendel's hypotheses. Specifically, I'm going to cover incomplete dominance and codominance. So let's look at these two tulips. We have a red tulip and a white tulip. According to Mendel's laws, there should be two alleles, a dominant uppercase allele and a recessive lowercase allele. There should be three genotypes, homozygous dominant, heterozygous, and homozygous recessive. There should be two phenotypes, red and white, and that's what we see here. In this case, the dominant allele is always expressed. So this red flower would either be homozygous dominant or a heterozygote. The recessive allele is only expressed in the absence of the dominant allele. Because the dominant allele overshadows the recessive allele, that means that this white tulip has to be homozygous recessive. So Mendel came up with four hypotheses, and one of them was that alleles have a dominant recessive relationship. And that means if you do a cross with a true breeding or homozygous dominant and a true breeding white or homozygous recessive, 100% of the offspring would be heterozygote and they would express the dominant phenotype. But there's exceptions to every rule. So sometimes scientists did a cross between the homozygous dominant and homozygous recessive, and they got a completely new phenotype. Instead of seeing the dominant phenotype expressed, they saw a new phenotype that was a blend between the dominant and the recessive phenotype. In this case, the new phenotype is pink. So what is happening here? And could this be due to a mutation? That's a pretty logical assumption here, and it's something that would definitely have to be tested. So how do we figure out if this phenotype is due to a mutation? Well, scientists did a cross between the two heterozygotes with the new phenotype. And if we do a Punnett square with what we would expect, and we put our alleles in the different boxes, the first box we would expect to be a homozygous dominant. If this was truly a mutation and the red allele was no longer able to produce red pigment but produced pink, you would expect that this would also be a pink flower. But in this case, scientists found that they got a red flower. The heterozygotes were of course pink, and then again for the homozygous recessive, they found the white flower. So this was not a mutation because both of the original phenotypes are present in the homozygous flowers when they did this cross. But there is this new phenotype in the heterozygote. So we have a new term, and that's called incomplete dominance. And that's when you have the appearance of a new third phenotype in the heterozygote. So in this case, you would still have two alleles, a dominant and a recessive. You would have three genotypes, homozygous dominant, heterozygous, and homozygous recessive, but you would have three phenotypes, red, pink, and white. The heterozygote phenotype, it's a blend between the dominant and the recessive phenotype. So in this case, the heterozygote is pink, which is a blend between the red and the white. There's another exception to the dominant recessive relationship, and that is exemplified by this cross here. We're again taking a homozygous dominant and a homozygous what we thought was recessive and ending up with a flower that has both phenotypes. It has red with the white stripes. So what is happening here? Well, scientists call this codominance. And that's where one allele is really not dominant over the other. They are both expressed. So although scientists thought that this would have been the dominant flower and this would have been the recessive flower, in this case, they are both dominant. In the heterozygote, both of them appear. So what's hard about this one, codominance, is the notation. So we have to do a different form of notation to indicate that the alleles are showing codominance. So what scientists have come up with is what we do to show each individual allele is for two alleles of the same gene that show codominance, we first have an uppercase letter, and it has to be the same letter. And you can choose any uppercase letter except I. I is reserved for blood type. So you need an uppercase letter that is the same, and that shows that they are alleles of the same gene. Then you need a superscript, and those superscript letters have to be uppercase. You can choose any letter, but they should be different from each other. And this is going to show that they're both dominant because both of these superscripts are in uppercase it shows that they're dominant because they're both sort of being held onto by this other letter that is in uppercase. It shows that they are alleles of the same gene. So in this case, our red flower would be indicated by an F superscript R, F superscript R, with both R's in uppercase. The white flower would be F superscript H, F superscript H, with the H being uppercase. And then the heterozygote that is showing both phenotypes is F superscript R, F superscript H. In this case, it's showing red with white stripes. However, any way that the offspring is showing both phenotypes would count. It could be red with white dots. It could have one white petal and one red petal. Any way that it is showing both phenotypes is an indication of codominance. So telling the difference between classic Mendelian inheritance, incomplete dominance, and codominance can be confusing for students. So I've got some examples going here. So we have our aliens. This is a green alien and a yellow alien. And what kind of things are we going to see if we do a mating with classic inheritance, incomplete dominance, or codominance? Well, in the case if we have a green mating with a yellow and we get all green offspring, this would be a case of classic Mendelian inheritance, where green is dominant over yellow, and then we could indicate the alleles with an uppercase G for green and a lowercase g for yellow. That means that my green parental would be homozygous dominant, GG. My other yellow parental would be homozygous dominant with lowercase g, and then my green offspring would be heterozygote. Let's look at another case where I have a green alien mating with a yellow alien and I get a pale green alien. That's a new phenotype, so anytime you see a completely new phenotype that's a blend between the two parental genotypes, you need to be thinking incomplete dominance. And this is a new phenotype that's a blend between the dominant and the recessive. So in this case, we could still use the uppercase G for green, the lowercase green G for yellow, so the parents would have the same genotypes as in the original cross. And the heterozygote would also have this same heterozygote genotype, but knowing that it shows incomplete dominance, it has a third phenotype. Let's look at one last example where I have my green alien crossed with my yellow alien, and I get an alien that is a different mix of both of these colors. This alien is expressing both the green and the yellow. It's not a blend, there's not a mix, but it's also clear that the green is not dominant over yellow because I have showing all of both of these phenotypes are available. This is an example of codominance where both the phenotypes are expressed together. So the hard part with this is the notation. So in this case, I chose an uppercase A to be my letter holding the superscript because A stands for alien. I chose a G for green and a Y for yellow. Again, the A, both of them having an A, shows that they are alleles of the same gene, and both of them having the uppercase superscript shows that they are codominant. So my green alien would be A superscript G, A superscript G. My yellow alien would be A superscript Y, A superscript Y. And my multicolored alien would be A superscript G, A superscript Y. So what you should know, if you're told that a gene expresses either incomplete dominance or codominance, you should be able to predict the phenotypes of the offspring. If you're given the results of a cross, you should be able to label the genes involved as either showing classic Mendelian inheritance, incomplete dominance, or codominance. And you should be able to use the correct notation for the different inheritance pattern. And I'm telling you now that most students have trouble with different notations once they have these exceptions. So be very careful and pay attention to detail when you're labeling the genotypes of your cross. That's all for tonight.